Hello everyone, Bismillahirrahmanirrahim. In the last lecture, we discussed about the exotic or heterologous gene expression overview, and we learned about the genetic code and what is codon degeneracy and what are the synonymous substitutions and codon usage bias between different organisms and rare codons. In this tutorial is in the continuation of the first lecture of the codon optimization. So for codon optimization, there are different factors, and we use different bacterial hosts. So which bacterial host should be, or microbial organism should be used as a host, or which is the good or better choice as a host for the synthesis of an exotic gene, or synthesis of a protein of interest. So it should be a host which should maximally support our protein of interest. So the choice of the host cell whose protein sensing machinery will produce the required or desired, or I would say that precious protein will initiate the outline of the whole process. So it defines the technology needed for the project, like how to say, like variety of molecules, different tools, equipments, or reagents. And among microorganisms, the host systems that are available include bacteria, yeast, the filamentous fungi, and unicellular algae. All of these bacterial hosts, you can say that, have their own weaknesses and strengths, and their choices may be subject to the protein of interest. But, however, till to date, it's one of my favorite hosts. There are certain reasons why it is my favorite host. But till to date, E. coli is the most preferred host which used in molecular biology. So therefore, in this tutorial, I will especially focus on E. coli. So the question is, why E. coli? Why E. coli? For example, for decades, E. coli is the first choice of, especially for expression host. Why? Because the advantage of using E. coli are well known. Or for example, they have certain advantages over other microorganisms, and I would like to discuss these advantages. Like it has unparalleled fast growing kinetics, means growth is like fast, it is as the fastest growth as compared to the other microorganisms. Like for example, in optimal growth conditions, doubling time is about 20 minutes. So it is the shortest doubling time for any bacteria, and because of that shortest multiplying time, therefore high density cell cultures can easily be achieved with E. coli. And other hand, the, we don't need such complex or expensive mediums. A rich complex media can be made from readily available and inexpensive components for E. coli to support its growth. And then the transformation with the exogenous DNA is also very important, that if you add certain plasmid or some gene you want to transform in E. coli, you can do it in as little as 5 minutes. For example, transformation can be done in E. coli as little as 5 minutes. And but the most important point is, the most importantly, the genetics of E. coli is well known and well characterized, and large number of genetic tool and parts are available for E. coli. Therefore, E. coli is the favorite host in molecular biology. However, there is, it's not so simple, not easy. So even after the careful selection of the plasmid factors and other tools and parts in E. coli, it cannot be predicted if the protein will be obtained in high amounts and in soluble active forms. So various situations that can impede reaching that goal can be encountered or faced, which unfortunately happens very often with E. coli. So there are different factors which can impede the function expression of desired protein in E. coli are the unique and different structure of the target gene, exotic gene, and the E. coli genetic setup. So exotic genes have different structure and E. coli has the different structure. And then the low stability of the RNA, especially messenger RNA, low translation efficiency, uneasy or improper protein folding, target protein degradation by E. coli proteases, and most importantly or most significant factor is the different codon usage between the E. coli and the foreign or the exotic gene, which can impede or hamper the soluble protein expression in high amounts. So, to make it further clear about difference in codon usage, I will go to the previous slide. Here you can see the usage of 6 least used codons in E. coli compared to the other organisms. For example, arginine, most, or we can say proline, that the proline frequency in E. coli of these codons triple C, CCC. When there is proline, E. coli prefer this CCC only out of 100, only 4.3 times. But on the other hand, Homo sapiens, humans, when the proline, they want to insert proline and they prefer these codons 20 times. So if we insert some gene in E. coli from the Homo sapiens and transform in E. coli, whenever the proline comes, it will search for the triple C in E. coli. But E. coli has very low pool of proline, triple CC. So when after this, when 4.3 of these codons after use, the Homo sapien genes will start searching for this codon, but it is not available in E. coli. So when there is no codon is available, so the protein machinery will be stopped, or other it can incorporate some uh, wrong codon in the protein sequence. So it results in a truncated form of a protein or low expression of a protein, or the protein will further not express, so expression will be low. So this is the preference of every host or every species for certain amino acids or codons. So it is the problem of the codon usage bias between different organisms, and it affects the expression of our it can strongly affect the expression of our foreign protein in E. coli, especially if we are inserting genes from some other organisms rather than E. coli. Even from different bacteria have different choices or preferences for different codons. So it generates different preference for different. It generates some discrepancy, and discrepancy leads to the low expression of the protein, our desired protein. So there are another terms we encountered when we are studying about the codons, rare and low usage codons. So there are, it can be we will define that what is rare and what is low usage codon. In E. coli, a low usage codon is defined as a codon that is used rarely or infrequently in the genome, with usage frequency lower than the smallest value among the usage frequency of the non-degenerate codons like methionine, AUG, and tryptophan, UGG. And the optimal codons for amino acids like leucine, valine, serine, proline, threonine, alanine, and few others that have two or more degenerate codons, with each having specific corresponding cognate transfer RNA for the optimal codons. On the other hand, the rare codon is infrequent codon or a minor codon is equivalently can be defined as a synonymous codon or a stop codon that is not only used rarely or infrequently in a genome but also decoded by a low abundant transfer RNAs. So the translation rate for a sense rare codon is much lower as expected than that for a common major codon due to transferability. This means that if some codon is rare, ultimately is cognate or corresponding transfer RNA also will be present in rare amounts. So rare the codon mean rare the transfer RNA is. Low usage, the one thing between the low usage and rare codon, you should know that low usage codon is not necessarily a rare codon like cysteine codon, UGC, threonine codon, ACU, etc. are not rare codons of E. coli. However, a rare codon is definitely a low usage codon. Mean okay, low usage codon is not always a rare codon. Some codons are present there in ample amounts in bacteria, E. coli pool inside flowers, but maybe it don't prefer those codons, so they use in that codons in low amounts. But on the other hand, some codons are present in very rare amount and bacteria E. coli use them rarely. Those are called rare codons. So low usage codon is not necessarily a rare codon, but rare codon is always, almost always a low usage codon. It means that bacteria, even some if codons are present in an ample amount, still it has some preference for some codons due as the number of factors which I mentioned in evolutionary factors, result of evolution, there the factors I explained in my previous lectures, which are supposed to be responsible for this preference, take which if can affect this is have has developed because that affects they have developed some preference for the certain type of codons for protein synthesis. So if we express the effect of rare codon or what are the effect of rare codon high level expression of heterologous protein, as I mentioned previous in this previous slide, that a number of studies have revealed that rare codons are capable of qualitatively and quantitatively causing expression problem in E. coli or other organisms, and these problems mainly occur on translation level rather than on transcription because the translation level because the amino acid codons are present in different numbers like low amount in high amount rare or this can affect the heterologous expression of the protein. For example, when there is codon biasness in your foreign gene and E. coli, so the ribosome can pause upon encountering a rare codon. It will stop and search for codon because that codon is present rarely in E. coli, but maybe in the parent strain from which that gene is derived, that is abundant. So it search for that, but in E. coli it's rare. So it will can if it ribosome stops for a longer time on messenger RNA, it can ultimately detach, or it can otherwise it can insert some wrong basis. So, for example, in so also so if the rare codon are present, it means there is depletion of low abundance transfer RNAs, and this can lead to the, as I mentioned, that misincorporation, truncations, and this will ultimately affect the heterologous protein expression. So in this figure, you can see that if there is insufficient transfer RNA pool, it can lead to the translation stalling, premature translation, termination, translation frame shifting, and amino acid. For example, if there is less frequent or rare codon comes on while protein synthesis on ribosomes. Ribosomes stalls, and the process gets slow. But when there is abundant or frequently used codon came encountered, it moves fast. So this pausing can have some detrimental effect on protein expression like frame shifting, termination, muscle incorporation, and low expression levels. Because this is due to the difference in codon usage and which can impede the translation due to the demand of one or more transfer RNAs that may be rare or lacking in the population. So different studies have shown that it has a great effect that the rare codons have effect that it can reduce the translation rate of the target gene, and the expressed protein is in low or undetectable levels. Amino acids are misincorporated into the target protein, or truncated or amino acid deleted peptide or proteins are synthesized, and frame shifting peptide or proteins are synthesized. So in E. coli, they found that the rare used sense codon is AGG for arginine. For example, in this table, they have mentioned the different amino acids, their corresponding codons, and the fraction in all genes and fraction in class 2. The class 2 is the fraction represented in 195 genes, highly and continuously expressed during exponential phase. So this is the fraction all genes and highly expressed genes. You can see both in fraction all genes, highly expressed genes, arginine of codons of these arginine are present in the less or rare amounts. You can see here that 0.003 is like arginine AGG, especially in highly expressed gene, is the low preferred or rare codon in E. coli. For example, if some gene we are going to express in E. coli, that gene that have more arginines and that have preference for AGG, that is maybe abundant in that organism. For example, in humans, for example, we can say like if genes we derive gene from human, and from human gene it maybe it has this gene present in higher amounts, rather in E. coli it is rare. It's every species, every organisms have a different preference. For example, rare in one may be abundant in other, or abundant in other organism may be rare in E. coli. So because of this discrepancy, all these problems caused by the rare codons in E. coli can happen. So codon biasness, another observation that this is species-specific biasness, and the, codon usage and transfer and abundance, heteroprotein expression is as a result. If there is this bias, is codon bias, the heterologous protein expression is often inefficient. So in summary, or in one line, we can say that rare codons reduce the protein expression. So now what's the solution of these? Sorry. So what are the solutions of this? Actually, there are two main solutions. Either because the rare codons have the rare transfer RNAs. So first one is express the rare transfer RNA to remove the bias in the host cell. Another is if it doesn't work, then we should go for the uh, gene synthesis. So how gene synthesis can work. In gene synthesis, we can replace the rare codon with the most preferred ones by using the phenomena of synonymous mutations, by the process of site-directed mutagenesis or de novo gene synthesis with codon optimization. So, in summary, we have the two procedures or techniques by which we can overcome this codon biasness. Is first the co-expression of the rare transfer RNA genes and the replace of the rare codons in gene with the synonymous optimal codons. Mm, several actually there are several laboratories have shown that expression yield of proteins whose gene contain rare codons is can be dramatically improved when the transfer RNA cognate or rare transfer RNA is increased within the host. So how you can, because every gene, every protein is coded by every protein, every transfer RNA or ribosomal RNA is ultimately coded by the DNA. So we can express these transfer RNAs in on a plasmid. For example, if E. coli has a 6 or 7 or 8 or whatever, they have the rare codons. We analyze that there are the rare codons. Scientists have developed some commercial uh, plasmids which can increase the amount of rare transfer RNA in very significant amounts. So can provide the uh, replace the rare codons with the can increase the copy number of the rare codon. So there will be no depletion of rare transfer RNA in these cases. So by own copy number can be increased. It depends upon the which plasmid vector you are choosing, high copy number, low copy number. Usually we insert the gene of these transfer RNAs on plasmid on a high copy number plasmid. So increase to produce the higher amount of the more the copies mean the more copies of gene mean more transfer RNA in the in E. coli pool. So it can solve the problem of the rare transfer RNAs present in E. coli. It's the first and the most easy process. So first of all, it is recommended to use this plasmid. This plasmid commercially available. Can use this plasmid. So these plasmids have different transfer RNAs and it can increase the translation efficiency of uh, the proteins. There were some other mutant strains specifically, they are specifically designed for optimal protein expression of the genes, especially the genes having the rare codons. And which can also commercial is it can also supplement the transfer RNA, and so we don't need to express the plasmid. Simply we can order the stain. The stain is specific design their genome. It don't contain plasmid. They have the inserted rare codons in its genome. So it is specifically designed stain because D3 stain is well known good stain for expression of the protein. So they developed the variant of this D3 and called it Rosita 2 or Rosita Gamma. It is recommended to first try this strategy. For example, either you should have the plasmid expressing the rare transfer RNAs, or you should have this stain because it's the most easy method and economical, very cheap, because synthesizing gene is, if you are for example in metabolic engineering, you have to synthesize like 15-20 genes. It is not economical, so it is a burden. So why not use this simple and cheap economical strategy? So it is first line recommendation. And if there is cellular expression and satisfaction irregular expression, then we can proceed with this. Otherwise it cannot correct, then we have no choice. other than to go for codon optimization. So in a summary, what is codon optimization? Codon optimization is like uh, it takes the advantage of degeneracy of the genetic codons or the by introducing the synonymous mutations that favor efficient soluble protein expression. So it will remove the rare codons with the codons, the most abundant codons in E. coli. For example, synthesizing artificial synthetic gene for codon optimization. In this figure, you can see that gene and this corresponding protein. The red sequences say, of red codons are the rare codons or rare codons. And in when we on online, there are certain specific tools when we optimize. It can give builds different builds in depend on 1, 2, 3, 4, 5, 6. We can select. According to our experience or knowledge, we can any of these 10 we can select, or any of the not stain, sorry, any of this build we can say. And for example, in this the build one, you can see that this the AGT is replaced by the AGC, which is the more abundant codon. And likewise TAA is like a rare codon is changed with the CTG, but the amino acid produced, the protein produced is the same. So it's not, it should not affect the amino acid. For example, if there's arginine, it should be arginine even after the change of the nucleotide in the codon. So the amino acid sequence should be similar, but it should be replaced by the most abundant codons. So introducing synonymous mutations that favor efficient soluble protein expression. It is the principle of this technique. So once you got the codon optimization, you get the after codon optimization, you select some gene and then you synthesize artificially. Then we clone this gene into our expression host, and then we evaluate. And if this is we get our corresponding band, then we can go for scale of expression, purification, and different. We can go for any purification. We can we're going to express this after purification. We can use it as a drug probe, or if we want to study three-dimensional structure whatsoever. Uh, Actually, there are still many features to consider when designing a full gene, which includes codon abundance relative to the cognate transfer RNA abundance, repetitive sequences or restriction sites. There should not be restriction sites, or we should know the sequence prone to uh, create secondary structures in RNA transcript. There should, when designing, artificially designing or de novo synthesis of the gene, a synthetic gene, we should consider also the repetitive sequence restriction sites, or it should not have secondary structures in RNA transcripts. So as you might imagine, it is not easy for humans to balance all these factors on their own, but luckily, many researchers have created codon optimization algorithms, and DNA synthesis companies such as IDT and GeneScript host online codon optimization tools. These tools are very user friendly and easy. For example, if you open this website of genes codon analysis tool, I mentioned this website here. Just open this and then you can select which host you're going to express your gene. You should know the sequence of your gene. What is the sequence of your gene? Then which host it give the different host. You can select the E. coli, and then you just simple paste your sequence here, and then analysis. For example, here I put some sequences from some bacteria. Even it maybe it is from Pseudomonas, maybe, yes, I think. So then we analyze. So it gives different parameters. It analyzes based on different algorithms. The first one, it gives numerical value. It's called codon adaptation index. So what is codon adaptation index? Very simply, we can say that the codon adaptation index is the most widespread technique for analyzing codon usage bias. The codon adaptation index is the actually very simple and easy. The codon adaptation index is a measure of the synonymous codon usage of a gene and the synonymous codon frequency of a reference set. So your gene and your bacteria, so it analyze the measure the synonymous codon usage between your gene of interest and your bacterial genome. So they say that if your value codon adaptation value is, for example, more than zero point 0.8. Yes, so it means greater, it should be codon adaptation index value should be, it is zero point 0.8. So near to 1 is ideal. So but it should be greater than zero point 0.8. Even if I take the sequence from the pseudomonas in E. coli, that equilibration index is 0.76, which is less than zero point 0.8. So it if even if I express going to express gene from pseudomonas, I need to optimize this gene for expression in E. coli for the optimal expression. And the GC content is from the GC content. If there is more AT rich sequences, it means that it is not stable because GC content should be between 30 to 70%. So it has the good bonding between G and C has the three hydrogen bonds. So the binding is good. They are more stable as compared to the ATC. So another is frequency, codon frequency distribution. Actually, they have the parameters. They just you don't need how they perform this, all this. They have different algorithms, but they mention that your gene should be have this frequency codon distribution between this value and this value. Is ideal, and with the chances that expression if this their codon frequency distribution is less than this value, it means the chances of your expression is low. Here, a graph of codon frequency distribution is is plotted to identify the quantities of rare codon present in the desired protein. Frequency of a codon usage with the value of 100. You can say that frequency of codon usage with a value of 100 indicates that the codons are highly used for a given amino acid. Conversely, the frequency of codon usage with a value of less than 30 is determined as low frequency codons, which is likely to affect the expression efficiency. For example, if there is there are only four codons which are ranged between the 21 to 30 and 31 to 40, but the most codons like 54 they have present in this 91 to 100 of range. So more the frequency distribution in the list of 90 to 100, it's more are the chances that the protein will be efficiently overexpressed. So after when they give first they give you the sequence and after analyzing they give you these values, for example, that your codon adaptation index is this, GC content is this content is anyway good. Then they give the codon frequency distribution. Then you can analyze, and then they give the option to optimize. So they will optimize for you and give you different builds with this kind of sequence. For example, before optimization, the codon adaptation index was 0.72, and now it after optimization codon index is improved. They have improved to 0.87. And GC percentage, average GC content was 50 and it is 56. It is good. And you can see that codon frequency distribution, it was like there are many between 51 to 60, 51, there are like 41 to 50, 31 to 40, 21. There are different codons which are falling behind the 50. So it remove those, replace those sequence as the result with the most abundant. So the frequency improved and all these become falls between 50 to 100%. So it from the average of 52 increase to 68. So it means the after this, if we use this build, and then we will, after we selecting this build, we order the company, so they synthesize these genes, like we synthesize primers, 1 kb 2 kb genes, and they have different price tags for we can synthesize, and they will send flies and then we clone these sequence, and we, with the hope that it will express with express in high amounts for our given gene. So this is uh, the method, one of the method which is the mostly used, because these tools, GeneScript and some other tools are available. The most widely used tool is GeneScript tool. This is how we can overcome the codon biasness. Thank you for watching.